Good afternoon. My name is Juan Mendez, and I'm going to attempt the Transforming Bacteria Simulation Lab. I'm going to start right now. So here are the predictions. It says the objective of the simulation is to transform bacteria with a recombinant plasmid using the heat shock technique. And I'm supposed to select the tube that best represents a successful transformation. I believe this will be tube 4. Because as you can see here, it contains both bacterium and plasmid cells. And I believe it's this one rather than this one, because here you can see the plasmid cells are already present in some of the bacterium cells, whereas here there are none inside. So I believe this is this one here. And this experiment, I'll have two samples, the control and the experimental. The control will be the P negative, and this will have E. coli cells, but no recombinant plasmid. And this is called a negative control because they will not be transformed due to the not having the presence of the plasmid. Unlike the experimental group P positive, the experimental group will contain both the E. coli and the recombinant plasmid. And we'll use the negative control to check for the bacterial viability and ensure that transformation has occurred in the experimental group by comparing both of them. And the tube that I think represents negative control outcome would be this one, because as mentioned above, this one has no plasmid cells, the recombinant plasmid, unlike tube 4. I'm gonna go through the protocol. I'm gonna set the water bath, add the component cells to both the P minus and the P plus tubes. And I'm gonna only add the recombinant plasmid to the P positive tube because that's the experimental group, and not to the negative one that has no plasmids. Then I'm gonna heat shock both, and then I'm gonna incubate the cells in the broth after transformation. So I'm gonna start with the steps. I'm gonna set the water bath to 42. Yeah. Then I'm gonna follow the instructions here. Pick up the P200 mic from the rack. I have it here. I need to adjust the volume to 50 microliters. Hopefully I can get it this turn. Yeah. Save volume. Now I'm gonna get a tip from the P200. Yeah, I already attached the tip. I'm gonna close the box. I'm gonna open the CC tube, which is here over at the ice bucket. I'm gonna move the micro pipette over here. Yeah, now I'm gonna drop to 50 of the cell solution by pushing the plunger for a stop and then releasing it. Yeah. Now I gotta open the B minus tube. I'm gonna move it over here, right? It's instruction. It says dispense the CC into the P minus tube by holding down the plunger until it releases the second stop. Okay, I'm gonna do that. The second stop. Okay. Place the tube in the ice bucket immediately. So I'm gonna put it right here. I'm gonna close it. Close it. And then I'm gonna open the biohazard container. I'm gonna dump the tip. And then I have to repeat this procedure. So I guess I have to add another tip. And then I gotta get some more CC. Then do the same thing over here to the plus. Yeah. I'm gonna put it here. I'm gonna close them. Yeah. So I'm gonna repeat the step. Check the tip. Close this. And then next, I'm gonna put this micropipette back where it was. And now it says pick up the P20 micropipette. So it's this one. I have it here. use the volume setting to change it to 10 microliters. So I'm gonna do that. Yeah, I'm gonna open the P20 tips and attach a tip just like the other one. Yeah, and then it says move the micropipette tip over the para R tube, which is this one. Yeah, and then it says drop to 10 the solution by pushing the plunger to the first step. Okay, so just like the first one, yeah. I'm gonna open the P plus tube and then I'm gonna dispense it. Gonna says I have to dispense it to the P by holding the plunger until each of the second step. Okay. Then I says mix the solution. Okay. And then put it back into the ice bucket immediately. Okay. And I'm gonna go back to the biohazard waste and eject the tip, close it. And then it says set the timer for 15 minutes by P plus. So it's okay. So I'm gonna go 15 minutes. Start. All right, perfect. So I'm gonna open the water bath lid, pick up the P minus and P positive tubes and place them into the floating tube rack. So here and here, yeah. Set the water bath timer for 45 seconds. But it's kind of sensitive, but yeah. Pick up the rack and put it in, okay. Close the water bath lid and start the water bath timer, which was 45 seconds. I'm gonna open the water bath lid, move the rack, move them back into the ice bucket. Yeah. Now set the timer for a minute. Yeah, set the timer for a minute. Start. Perfect. And now I gotta pick up the P200 micropipette again. I gotta change the volume to 150 now, apparently. So I'm gonna do that. Okay. Get it correct. Good. Yeah, perfect. I'm gonna open the tip box, attach a tip, then close the tip box, and then I gotta pick up the micropipette, place the micropipette so the tip is in the LB solution, the broth. Okay, perfect. So it's here. Yeah. Drop by pressing the plunger down to each of the first step and then slowly releasing it. Yeah. Now I'm gonna open the P minus tube. The P minus tube. I opened it. Dispense the broth into the P minus tube by holding on the plunger. Okay. Perfect. Mix the solution. Okay, and then I'm gonna open the biohazard waste. Then I'm gonna eject the tip. And then repeat the process to add 150 to the P tube. Yeah, to the P plus tube. Put it here. Then I'm gonna close this. Touch another tip. Yeah. Then I'm gonna extract this. I'm gonna put it in here. Perfect. And now I gotta close this and this. Gotta eject the tip. And then put this back. And then I gotta set the timer for 15 minutes and incubate the cells at room temperature. That's why I took them out of the ice bucket. So let's get 15 minutes. Perfect. All right, and that should be it. So the results, after you have performed your transformation, you will not be able to confirm by simply looking at the tubes. The next step is to place the bacteria in solid medium so that you can observe the growth. For the simulated environment, we have provided a hypothetical view of your results inside the tubes. So as I predicted, both of these are correct because the positive ones would have the bacterium cells with the plasmid cells already entering the bacterium, just shown as in the actual results from my predicted results. And well, the negative, we didn't have any of the plasmids present as predicted and shown with the actual results. So I'm really glad I got to get all of those correct. So let's see. A transformation was successful because some of the bacteria in the experimental group contain plasmids. The bacteria in the control group should have no plasmids, yeah, as I said before. Below is a representation of the ideal results after the transformation experiment. For successful transformation, the secret plasmid DNA needs to be taken, yeah, into the bacterial cells. And in the negative one, there's only E. coli cells, no plasmids, yeah, as I said before. So now I'm gonna go into the reflection. Let's look at what you learned from the simulation. Please click on your answer. What is bacterial transformation? A process through which genetic makeup of a cell is altered by introducing foreign DNA into bacterial cell. Yeah, this sounds like it. What is this one? See? A process in which the genetic makeup of a cell is mutated by introducing new enzymes and proteins. And not really. The last one, a process in which dead bacteria. No, it's not dead. So I assume it's this one. Well, I know it's not. We're not gonna assume stuff. This sounds the most correct. Yeah. How does the P negative tube differ from the P positive tube? Well, I'm just gonna read that. P negative has a controlled plasmid added to it that does not contain a gene for amplification resistance, while P minus doesn't have plasmid in it, so I don't think it's this one. P minus contains the recombinant plasmid, but no competent cells. No, P minus doesn't contain plasmid. P minus contains the competent cells, but no recombinant plasmid. Yeah, I think it's this one. Yeah, it was this one. Similarly, whether the following statement is true or false. Luria bras, the LB we used, is primarily used to culture or grow bacteria. Yeah, that's why I added it. Yeah, I believe it's true. Yeah, it was true. You used a septic technique during this lab. Why is it important to work in a sterile manner when working with bacteria in the lab? Let's see, to prevent DNA contamination from the environment that may also then be transformed into bacterial cells. Well, this sounds correct, yeah, but I can read the other ones. To prevent the transfer of bacteria from the experimental environment and vice versa. Yeah, this is true, this is true. And the last one, sterility is not important in this lab. It really is important because you can have contamination, so I believe it's this one. Yep. Why are the cells incubated at 42 degrees? At 42 degrees, bacterial cells begin releasing DNA for other cells to take up. Well, you're not releasing, you're taking DNA in as I understand it. This is a good recovery temperature after heat shock. Not really. At 42 degrees, the membranes of bacterial cells begin to degrade Creating pores through which DNA can enter, yeah, because DNA enters, not released. Yep, perfect. Feedback question. How confident do you feel about using technique? Well, I feel very confident. Thank you. Yep, so next is the summary, and yeah, that's about it for the lab. So thank you, and I was very glad to try this lab, Miss Arnold. So can't wait to see what else we will do in the future. Okay, I'll stop.